KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Now in this video we are discussing complete information of Anaphase. Okay, in Anaphase here we will discuss. Yes. So in this video complete information of Karyo Kinesis we will discuss. So cytokinesis in another video I will give you. Why? Because our knowledge point of view, this one Anaphase we can be divided into two tabs or two processes. One is a karyokinesis, means a division of nucleus, and the second one is cytokinesis, division of cytoplasm. We have seen now. So now under karyokinesis again 4 stages we can observe. So first one is prophase, metaphase and anaphase, and one more is telophase, isn't it? So there in a prophase what was happened or what is happening, so somewhat information I have given, but key points means what are the main important points to be learned or we have to remember for NEET examinations. So I have given these points here. So before entering into the actual concept, so here what is happening? So in entire mitotic division, so what are the main important processes taken place? So now with one small diagram I will explain. Now here this particular cell is a parental cell. So parental cell diploid number is equal to 10. Okay, so 10 number of chromosomes are there. After completion of mitotic division, one particular parent or mother cell is divided into two daughter cells. Okay, so two daughter cells there. So these two are daughter cells or offsprings. So even these offsprings also consist of, or these offsprings also having, so same parental number. So what is a parental number is present in so in a parent, same chromosomal number we can observe even in offsprings also or in daughter cells. So thereby this is the main important point we have to discuss or we have to remember more and more. Okay, suppose if one parental cell or if one particular cell undergo the process of mitotic division, means so from that particular cell definitely results in the formation of two offsprings or two daughter cells. Okay, and now these two daughter cells also so consist of same number or the same number of chromosomes. So what we have seen in parents. So likewise somewhat diagrammatical explanation here we can learn. Now in a karyokinesis our knowledge point of view, so there 4 different types of stages we can observe. So let us start to discuss. First one, prophase. So prophase is the first step. In a prophase, what are the main important points to be discussed? So first of all, first point, these are all key points. Now chromosomal material, important chromosomal material condenses to form compact mitotic chromosomes. So in the previous class we have learned, so what is the information we have learned? Chromosomal material condenses to form what? Compact mitotic chromosomes. Means here what is a thread-like chromatin material is there. So that particular thread-like chromatin material by the process of condensation form what? Compact mitotic chromosomes. So thereby here formation of chromosomes here we are observing in case of prophase. And moreover, these compact chromosomes or mitotic chromosomes are developed from the or formed from the thread-like chromatin material or chromosomal material. They have given chromatin material, so we can say. Next one, chromosomes are seen to be composed of two chromatids. So already I asked to remember this one. So don't forget this particular point in your mind. Okay, so now what is the point? Now chromosomes are seen to be composed of two chromatids, so which are attached together at the centromere or at yes. So means what are the chromosomes or what are the chromatids are present in the chromosome? No. So all these chromatids are definitely attached at the centromere. So thereby here we are learning one point. Chromosomes are seen to be composed of two chromatids attached together at the centromere. Okay, next one, centrosome. So centrosome means already we have seen in animal cells, definitely this particular centrosome organelle which helps in the formation of bipolar spindle spindle fibers, likewise we see no. So likewise this particular centrosome which had undergone the duplication during interphase begins to move towards the opposite poles of the cell. So now in interphase, what is the information we are discussing? Centrosome which had been undergone duplication during interphase. Actually, the duplication of a centrosome we are observing in case of interphase only. But here we are understanding concept point of view, we are taking under prophase. So thereby when these particular centrosome undergone for the duplication only in case of or in the division of a interphase, okay, so begins to move towards the opposite poles of the cell. So means duplication process or the duplication of a centrosome taking place in interphase itself, but started to move towards the opposite poles, that particular process taking place in prophase. Point purida. Ipo centrosome irikama, ade centrosome duplication we have seen in duplication multiplication one number rendu nalu and the madari. Okay, so which one centrosome? Ade duplication we have seen in case of interphase da, but that particular centrosomes started to move towards the opposite poles of the cell in case of prophase. Okay, now what is happening now? Each centrosome radiates out. So definitely forms the microtubules called asters in case of animal cells. So there each centrosome radiates out microtubules, means from the centrosome to microtubules will come out as or some kind of tube-like structures known as asters. Some kind of structures like asters, or these kind of microtubules only we will be taking as asters. Okay, now the two asters together with spindle fibers forms mitotic apparatus. Means here if you see the mitotic apparatus or the spindle fibers of formation, if you observe there, no, in interphase a centrosome duplication process takes place. So these centrosome in a prophase start to move towards the opposite poles. Once these centrosome moves to the opposite poles, now here, so these astral this centrosome forms the microtubule at each polar region. So these microtubules only we will be calling as astral rays. So these asters, so means two asters we have taken two means sarangiriga means. So these two are present on both sides or both opposite poles. Now comes together with and form mitotic apparatus. Mitotic apparatus. So definitely mitotic apparatus formation point of view, definitely there is a involvement of centrosome only in case of animals. So plants point of view means bipolar spindle fibers of formation takes place, and these are also formed from the microtubules. So definitely with the involvement of tubulin protein, microtubules of formation takes place, and finally bipolar spindle fiber formation takes place in case of plants. Next one, cells at the end of prophase. One more time, cells at the end of prophase. When we view when we viewed under microscope, no, do not show Golgi complex, endoplasmic reticulum, nucleolus and nuclear membrane. So thereby here one point we can understand, even though we are taking eukaryotic cell for the study of cell cycle and cell division, so by the end of the prophase, at the end of the prophase, no, we could not find the presence of these kind of cell organelles in the cell. So thereby here the Golgi complex is absent, endoplasmic reticulum will not be appeared, nucleolus will not be appeared, likewise nuclear membrane also even will not be appeared in the end of the prophase. So this is a small information regarding prophase 1. Now let us start to discuss the metaphase. Okay, so right. So due to the disintegration of a nuclear envelope, chromosomes are spread throughout the cytoplasm of the cell. So here we have seen, so nuclear membrane, everything disappeared in a prophase or end of the prophase, we see no. So thereby due to the disintegration of nuclear envelope, so what is happening? Chromosomes are spread throughout the cytoplasm. Now there is no nuclear membrane. So thereby what are the chromosomes are present in the nucleus? Due to the absence of nuclear membrane, all chromosomes are now transferred to the cytoplasm of the cell. Point puri the cytoplasm of the cell. So by this time condensation of the chromosomes is completed and they can be observed clearly under the microscope. So in this particular case, no, metaphase. So metaphase definitely helps or definitely gives the like a conspicuous nature. Okay, means here very clearly we can observe the structure of chromosomes in the metaphase. That's why suppose this particular point you have to remember. So prophase is there, metaphase is there, anaphase is there, and telophase also is there. Now one point you are learning, out of these 4 stages, where we can study or where and which stage we can observe the study of or we can observe the proper structure of chromosome, means our answer must be metaphase. Why? Because in a metaphase, so what is happening? Condensation of the chromosomes, means the chromosomal condensation, everything will be completed and that particular chromosome observed very clearly under the microscope. Means here in this particular case the morphology of the chromosome, so morphology of the chromosome we can observe very clearly in the metaphase. So that's why I think so that particular point also I have written. So once let us search for it. Next one, and they can be observed clearly under the microscope. In this stage, morphology of chromosomes is mostly easily studied. So we can study about the morphology of the chromosomes very clearly. Next one, on the both sides of the centrosome. On the both sides of the centromere, we are finding the presence of kinetochores. Here, what is information we have written here? On the both sides of a centromere, 2 small disc-shaped structures known as kinetochores helps in the attachment of bipolar spindle fibers or simply spindle fibers. So thereby on both sides of the centromere, one diagram we have shown no. So on both sides of the centromere, so whatever the structures are there, disc shaped structures are present. These disc shaped structures only we are calling as kinetochores. These kinetochores helps in attachment of spindle fibers. So these spindle fibers will go and binds at the kinetochore region. So that is a main important point we can be discussed under the metaphase. Now main important characteristics or key points or the key events of metaphase. Now let us see. So first one is spindle fibers attached to the kinetochores of the chromosomes. First point. So here the first point is the spindle fibers are attached to the kinetochores of the chromosomes. Means here what are the spindle fibers are there? No, all these spindle fibers so definitely attached to the kinetochore regions. So kinetochore region in metaphase. So later chromosomes are moved to spindle equator and get aligned along the metaphase plate through the spindle fibers, so through spindle fibers to the both poles. So thereby here, what is the point here we are understanding now? When spindle fibers, listen carefully, when spindle fibers attached at the both regions or attached at the kinetochore regions simply no, so thereby, so these particular chromosomes start to move towards the spindle equator, or simply so moving towards the equator place, or equator means where we can find out no, exactly at the center of the cell, and get aligned along the metaphase plate. So means on the metaphase plate here, whichever formation takes place, the all chromosomes will come and sit at the metaphase plate or equatorial plate. So that is a main important situation regarding metaphase. So in metaphase, what are the main important points to be discussed means, so chromosomes are what are the kinetochores are there on the chromosomes, no? These kinetochores are attached by the bipolar spindle fibers. So first point. Next point is, so what are the chromosomes are present or situated on the cytoplasm. So all these are all started to move towards the equator place and get aligned at the metaphase plate. Okay, so when these chromosomes are aligned at the metaphase plate, no, automatically now at the time, no, all chromosomes are seems to be in one line. So that particular phenomenon we can observe in case of metaphase. Okay, next one is anaphase. Now third stage under karyokinesis, anaphase. So in anaphase, what are the main important points? No, each chromosome arranged at the metaphase plate is split simultaneously, and two daughter chromatids are now referred to as daughter chromosomes of the future daughter nuclei. So now in anaphase, what is happening? In anaphase, what is happening now? So each chromosome arranged at the equator plate is split. Means in anaphase, first point, what is the point we are discussing now? In anaphase, what are the chromosomes are present at the equatorial plane? In anaphase, such particular chromosome get split. If once you come down and you see the diagram here, so here suppose one particular chromosome here we have taken. So exactly at the spotlight now centromere on both kinetochores are there. Now due to the, so these are all bipolar spindle fibers. So here due to the condensation or due to the pulling and pushing mechanism, so thereby these pulling of bipolar spindle fibers from the opposite poles, now automatically one particular chromosome, this diagrammatically I have drawn. So this particular chromosome split through the centromere or splits along with the centromere. So thereby the one part one side and the remaining part that side it will move. So that's why here, what is the point here we have written? Each chromosome arranged at the metaphase plate is split and simultaneously, split simultaneously, and two daughter chromatids now referred to as, so when one particular chromosome is get splitted, now that particular chromosome we will be taking as a daughter chromosome. So now that particular daughter chromosome is of the future daughter nuclei. So means in a future, so when that particular nucleus later means after completion of anaphase, in a telophase, no reappearance of nuclear membrane process we will observe no at that time. So definitely, so all these kind of daughter nuclei again surrounded by the nuclear membrane. So thereby here this particular point we are discussing. So in anaphase 1, so what are the main important points here we have to discuss means, first of all, once you see in key events we have written, so centromere splits, not only chromosome, even centromere also gets split. Okay, centromere splits and chromatids separate. Now the separation of chromatids process takes place by splitting of the centromere. Okay, now chromatids move towards the opposite poles. So thereby definitely what are the chromatids are there? Now all these chromatids start to move towards the opposite poles. So next one is so now these two are key points under anaphase. Key points under anaphase. Next one, in a telophase point of view. In a telophase, what are the main important points are there? Means it is a final stage of karyokinesis. First point itself important. So prophase one is a first stage of karyokinesis and telophase is a final stage of karyokinesis. Now in a telophase, what is happening now? Let us see once. Now the chromosomes that have reached their respective poles, so means here due to the pulling of bipolar spindle fibers, so automatically what is the process we are observing? The chromosomes that have been reached their respective poles. Now these chromosomes means like a daughter chromatids are there, no, or a daughter chromosome. These two daughter chromosomes mean one particular chromosome is splitted into two daughter chromosomes, isn't it? So these two daughter chromosomes start to move towards the opposite poles in anaphase. In a telophase, finally, so these daughter chromatids are reached to the respective poles. Okay, so respective poles. Now by the process of decondensation, so again chromosomes are now converted into thread-like materials. A point you understood. So thereby, in prophase, no, we have seen in prophase we have seen what is the thing. So by the process of condensation, thread-like chromatin material condense and converted into chromosomes. We have seen, no, in anaphase what is happening, no, chromosomes by the process of decondensation now converted into thread-like material. A point understood. One more time, in prophase, no, we have seen thread-like material by the process of condensation converted into chromosomes we seen. In anaphase, what is the thing? By the process of decondensation, chromosome converted into thread-like material. So rendume like opposite. That's why some cases, no, we will learn like this. Prophase characteristics are opposite to the telophase, or telophase characteristics opposite to the prophase. Point you understood. So thereby in a telophase what is happening? So by the process of decondensation, chromosomes get converted into thread-like materials. So thereby what is happening? Lose their individuality. So finally, lose their individuality, finally thread-like material formation takes place. That particular thread-like material only, our subject point of view, we are calling as chromatin material. Now key points or key events under telophase, once let us focus. Now chromosomes cluster. So chromosomes cluster at opposite spindle poles. Chromosomal cluster at opposite spindle poles, and their identity is lost as discrete elements. So once the chromosomes are reached at the opposite poles or reached to the opposite poles, their identity is lost. Okay, so the identity is lost as discrete elements. Next one, nuclear envelope. Nuclear envelope develops around the chromosome clusters. One more time you see there, nuclear envelope develops around the chromosomal cluster at the each pole, forming two daughter nuclei. So thereby on both poles, no, because of the reformation, because of the formation or because of the reformation also we can. Okay, because of the formation of nuclear membrane, what is happening now at both polar regions, no, two daughter nuclei formation takes place. So now here, if you see that prophase, no, there we have seen disappearance of nuclear membrane we have seen. But in a telophase, no, what is the thing we are observing? Nuclear envelope develops, means reappearance we are observing. So when nuclear membrane reappears or the development process is going on around the chromosomal cluster, no, automatically finally leads to the formation of two daughter nuclei. Simple only. Next one, finally, so in a telophase, nucleolus, Golgi complex and endoplasmic reticulum reformation process also takes place. Nucleolus, Golgi complex and endoplasmic reticulum reform. So means up to here means already these kind of cellular organelles disappearance we have seen in prophase. So once the nuclear divisions everything is completed, means once the karyokinesis is completed by the telophase, now again we are observing the reappearance of nucleolus, Golgi complex and endoplasmic reticulum. So here diagrammatical explanation, also small diagrammatical explanation I can give you. So this is plant cell, so there so this is nuclear membrane. So nuclear membrane, so within the nuclear membrane chromatin material, thread like chromatin material it is. Thread like chromatin material. Now in a prophase, what is happening? By the process of condensation, this LMA, thread like chromatin material converted into chromosomes. Now due to the disappearance of nuclear membrane, all chromosomes are scattered in the cytoplasm. So this entire liquid is a cytoplasm only now. So now all chromosomes are present in the cytoplasm, scattered. Next one, in metaphase, what is happening? What are the scattered chromosomes are there? All are arranged at the equatorial plate or metaphase plate. Equatorial plate or metaphase plate, and each and every chromosome. So here diagrammatically separately I have given the structure of chromosome there. What is information? No. Suppose if you take one particular chromosome, here exactly at spotlight we will see the presence of centromere. On both sides now we will observe the disc shaped structures known as kinetochores, and if these kind of bipolar spindle fibers are there now, all these bipolar spindle fibers will command attached at the kinetochore region. So these are all bipolar spindle fibers. Bipolar spindle fibers, one bipolar. Bi means two. Means what are the microtubules? What are the spindle fibers which are coming from the both polar regions? No, simply we will be calling as bipolar spindle fibers. Now attached like this at the equatorial plane. In anaphase, what is happening? When these bipolar spindle fibers pulls the chromosomes, no, that particular chromosome, each and every chromosome splits like this. So chromosome split. So each and every chromosome splits like this, like thereby here this particular chromosome splits along with the centromere. Along with the centromere. Now here this particular process. So, and these chromosomes start to move towards the opposite poles. Start to move towards the opposite poles in case of anaphase. Next one, in a telophase, what is information here we can understand now. In a telophase, now only we have seen now, due to the decondensation process, thread like chromatin material, so by the process of decondensation, chromosomes now get converted into thread like chromatin material. Later, around of this thread like chromatin material, so nuclear membrane reappearance or reappearance under nucleolus, Golgi complex and endoplasmic reticulum reformation. So now reappearance of nuclear membrane, and here under nucleolus, so here a small dot is there, nucleolus formation, Golgi complex formation, and endoplasmic reticulum likewise all kind of cellular organelles formation we will observe in case of telophase. So thereby here what is information? Sequential study. Okay, first of all prophase, next one is metaphase, next one is anaphase, and final one is telophase. So up to here we have studied exclusive study on karyokinesis. Okay, next one, in next video I will give you information regarding cytokinesis and significance of mitotic division also we will study. Okay, so this is a small information regarding karyokinesis. In the next video, let us see cytokinesis. Thank you.